Great. Yeah, thank you so much for having me. It's really great to be sharing this time with luminaries in the field like you, Shan, and others. So thank you again for the opportunity. I thought I would tell you a little bit about today some of the efforts that we've been doing in the last couple of years, and it's mostly around using computational methods for drug discovery, and more specifically, like the title says, for antibiotic discovery. And you know, drugs are incredibly slow and expensive to develop, like it's been mentioned. From the moment that you discover a new drug in the laboratory that looks promising to the time where that drug is going to have an impact on patients, it takes, on average, over 10 years. So it's a very long and windy road. And the cost associated with that process is immense. So it's, it says here around $2.6 billion to develop a drug. And I like the comparison of this. So the NASA has a budget to take a rocket all the way to the moon that is actually inferior to this budget, and so it's actually in some ways it's more expensive, and in some ways more of a huge deal to develop a drug than to even take a rocket all the way to the moon. And so, but the good news is that we've experienced a number of trends over the last decades that in my lab we think, and others think, that could help address some of these gaps. The first one is the ever increasing compute power that we have. This is obviously following Moore's law with the number of transistors that you can fit in a chip, which basically is doubling continuously over time. And this has allowed us to now have computers that can process huge amounts of information. And the other trend is data availability, right? Through advances in automation and advances in high throughput assay development, we can now generate more data than ever before. And so we think that combining these two things, we can help reduce the time that it takes to discover new drugs, and we can help reduce the cost associated with this process. And in particular, we focus on antibiotics and antibiotic resistance. And the motivation behind this is that this is a huge global health problem. The current predictions is that by the year 2050, around 10 million people will die every year in the world as a consequence of untreatable infections. This, as we can see here, will surpass every other major cause of death in our society. So we're really facing a critical time now, where we really need to come up with new antimicrobial strategies to help us sort of avoid this really sort of depressing future. And obviously, antibiotics are not only helpful when we have an infection. You take the antibiotic and they cure you completely. They treat the infection. But antibiotics are also essential for contemporary medicine. So things like childbirth or chemotherapy treatments, organ transplantation, surgeries would not be the same without effective antibiotics. And so they really permeate every layer of contemporary medicine. And actually, these 10 million deaths per year, that corresponds to the staggering number of one death every 3 seconds. And so really, it's an urgent problem. And I'm showing a photo of Earth here because basically every major antibiotic that we have available today, it comes from nature. It comes from the biological world. And nature, since the discovery of the very first antibiotic, which was penicillin, which was discovered by Alexander Fleming in 1928, nature has been a great inspiration as a source of antibiotics. And here I'm showing some of those chemistries that have saved millions of lives in the world that we have used, as life-saving drugs. But the problem is that for decades now, we have been unable to find new chemistries in biology that allow us to kill bacteria effectively. And so what we're trying to do instead of relying on nature completely, we're trying to encode the chemical complexity of molecules into the binary code of ones and zeros, so computers can take care of the discovery process. And in particular, in my lab, we focus on peptide molecules, similar to Yushan. We so far have mostly focused on linear peptides, though. We've done a couple of projects on cyclic and constrained peptides, but this is what we're trying to do. And Of course, as has been mentioned before, we follow basic rules of protein and peptide engineering that the structure of a protein or a peptide determines its function. And so if we can control the sequence of amino acids, the building blocks, we can control their activity. And this is a little bit like playing Lego, right? And so today I'm going to tell you about some efforts where we're using computational approaches to try to program peptide molecules. And there are several things that computers can help us do. They can help us explore sequence space, which is incredibly vast for any molecule, but also for peptides or proteins. It can help us generate new molecules, and this is we enter a little bit into the realm of creativity that we characteristically associate with the human brain, but computers are starting to make some advances in creating new things as well, although we're still learning how to do that. And then finally, computers can help us mine biology to try to discover novel antibiotic entities or novel potential drugs. So I'll start with the first one. How do we explore sequence space, and how big is huge, right? So I'm going to show here combinatorial sequence spaces of things that are concepts that may be more familiar to the audience, like number of people on Earth is around 10 to the 10. The number of bacteria in a human, we're surrounded by microbes, is 10 to the 13. The number of stars in the universe is 10 to the 31, right? So almost unimaginable numbers, very difficult to grasp. And now if you consider a very small peptide composed of only 25 amino acids on a linear chain, so again, very small, smaller than any protein in our bodies, which is typically composed of hundreds of amino acids. That very small molecule gives us a sequence space that is superior to the number of stars in the universe. And so we clearly need computers to explore this sort of explosive combinatorial space of molecules. And to make things even more complex and therefore more interesting from a scientific perspective, through billions of years of evolution, every molecule that has been explored throughout the evolutionary process, we can fit it into this like little pink oval, and all these white areas that I'm showing here, they remain unexplored. And so, what machines can help us do is also expound the boundaries of sequence space to try to reach some of these white areas of unexplored unexplored sequence space that may yield peptides or different molecules that can help us solve present-day problems, such as, for example, antibiotic resistance. So that's kind of the hypothesis that we have. And so we clearly needed computers to approach this problem or set of problems. And then the next questions that we asked ourselves was, how can we teach a computer to innovate at the molecular level to create diversity? And we reached the conclusion that the best way to do this was to train the computer to essentially mimic the greatest engine for diversity generation that we have, and that's nothing other than evolution itself. And so we decided to teach computers how to execute the theory of natural selection from Charles Darwin. So through evolutionary programming. And here's essentially how we did it. We encoded an initial population of peptides from nature onto the computer, and then we taught the computer how to evolve them through mutation, selection, recombination, and n-fold iterations of that process in a feedback loop. So essentially, the computer is evolving the molecules in real time in silico. And here we can see an example of different iterations and how the computer is evolving the molecules towards optimal fitness values that correlate with predicted antibiotic activity. So at least in principle, the computer is making the molecules better, optimizing them for antibiotic activity. And we follow this fitness function is what drives the whole evolutionary process, and I can get into details in the Q&A. And in the process of doing this, the algorithm is also capable of exploring previously unexplored regions of sequence space. So some of the white areas that I mentioned a couple of slides ago, yielding molecules with amino acid compositions that are atypical, not what we typically find in biology. So here I'm just showing some of the molecules that the computer generated, some of the peptides. We can see the beautiful alpha helical structures. But here, of course, we reach a roadblock that is common to any projects involving computers. And that is that everything that we see on the screen is just is based on statistical assumptions from the computer standpoint, that the computer thinks that these molecules will be great at killing bacteria, but we still need to validate everything, right? We're not at the stage in this field where we can just blindly rely on the computational assumptions and trust them. And so what we did here, we synthesized a lot of these using chemical methods, using solid phase chemical synthesis, and we tested them in a number of screens against real bacteria from laboratory strains and clinically relevant strains. We're able to identify that this molecule, wabanin-2, was actually the best one at killing bacteria with a relatively potent antimicrobial activity. And this is the three-dimensional structure of wabanin-2 as determined by NMR. And here we have the same molecule in 3D space. And we're not only interested in creating new interesting peptides and testing them in vitro, but we're also very much fascinated by the possibility that some of these molecules might have might be clinically relevant, or at least have some sort of translational potential. And so we decided to test this peptide in a mouse model, a skin infection model. And here we essentially infected the animals, and then we treated them with wabanin-2, the machine-made peptide, and two of the templates that the computer started the optimization process with. And here we have the untreated control group of mice, so you get a sense of how many bacteria are in the mice. So we can see that treatment with the synthetic peptide reduced the infections much better than the other groups that we had. So anyway, this was exciting to us, and it opened new avenues in our minds to using computational tools in general to be able to design novel antibiotics that were not only like really nice to look at on the screen, but actually some of them were capable of killing bacteria in vitro, in petri dishes, and also in preclinical mouse models. So just briefly, I'll tell you about another story here where, in this case, we identified this pentapeptide motif colored in red here that was conserved in a bunch of proteomes, and it was associated with a dual activity, antimicrobial and immunomodulatory activity. So the basis for this project is we thought, okay, can we just play Lego here? Can we stitch this pentapeptide motif onto a different molecule to endow that different molecule with those two properties that we want? So we took this peptide here, which is actually a toxin, and we incorporated onto its N-terminus the pentapeptide motif that we see here to generate the synthetic version of the molecule. And long story short, we were able to show in vitro and in vivo that this synthetic version actually was capable of resolving infections through the conventional means, which is direct antimicrobial activity, meaning the peptide is targeting the microbe and killing it directly, but also indirectly by boosting the host's own immune system, in this case, the mouse immune system. So this is just introduces a new paradigm for treating bacterial infections. Of course, immunotherapies are very common approaches for cancer treatment, but really, and for viral treatments as well. But in the case of bacterial infections, they haven't really been implemented very much. And so this opens up new avenues for thinking about how to treat bacterial infections in different ways. And we also showed that this dual sort of synthetic peptide with dual properties was capable of conferring protection against otherwise lethal infections in a sepsis mouse model, both against laboratory strains and against clinically relevant drug-resistant strains. And so this obviously sepsis is a huge problem, as I mentioned here. It kills many people per year, and so these were also encouraging results. Okay, so I've told you how we can use computers to help us design novel antibiotics, and next I'd like to tell you about how we can use computational tools to be able to discover novel antibiotics in biology or in biological data. And here we take a little bit of inspiration from algorithms that everybody here uses, right? Pattern recognition algorithms for facial and speech recognition. But here, instead of recognizing faces or sounds, what we want to recognize is amino acid patterns, right, that are encoded in proteins that might represent potential antimicrobial molecules. And the analogy that I like to use here is like if you had a huge Word document and you wanted to find a particular word within the document, for example, I don't know, peptide. You would go to the search function in Word, you would type peptide, and then the algorithm, right, it would identify it in every it would identify that word peptide in every instance that it appears throughout the document. So these algorithms operate in a similar fashion. We kind of know what we're looking for. So we type it, and then the algorithm, instead of searching through a Word document, it searches through genomes and proteomes to try to find potential peptide antibiotics. And to illustrate this concept in a more visual way, we can have a protein in 3 dimensions, right? We can display it in 2 dimensions with the amino acid code. And the algorithm, what it does is it essentially runs through the code, and it identifies regions, such as these regions in the different colors, that constitute potential antibiotics. And again, only a prediction, but it's a very powerful one because it allows you to explore entire proteins and then identify regions, such as this one in yellow, that might represent a novel antibiotic, and then we can extract it and play around with it in the laboratory. So using an algorithm similar to this, we're able to do the first proteome-wide exploration of the human body, searching for peptide antibiotics, and we were able to find thousands of what we call encrypted peptides. And the really interesting thing is that we didn't only find them in the innate immune system, which is where you would find antimicrobial molecules, right? The innate immune system is the one that help us fight off invading organisms when they take over. But we also found them in the nervous system, the cardiovascular system, the digestive system, you name it. And so we now have a hypothesis where we think that perhaps immunological response is not only a responsibility of the immune system, but we have all these other systems throughout our body that are cooperating either directly or indirectly to target infections or infectious agents. And I'm going to tell you briefly a couple of things that we found from this work. And so we found thousands of molecules. Then we synthesized 56 of them using, again, solid phase chemical synthesis. And we characterized them, right? We validated their antimicrobial activity in vitro. And then here I'm showing you how what we did here is synergistic interaction assays where we were wondering, right? If you take some of these encrypted molecules that are encoded in proteins that are in the same biogeographical area within the body, do they work in conjunction to target pathogens? And so for this, what we did is we combined them in cocktails. So let's say proteins that are produced in the same area of the body, the nervous system or the bloodstream, we bind them in cocktails. And what we found is that many of them were able to potentiate each other's activity to target pathogens. So that means lowering the concentration needed to kill bacteria to even nanomolar range when you combine them in cocktails. And we did combinations of 2 and 3 different cocktails. So this is interesting, right? From an evolutionary perspective, also thinking about, okay, if we have all these things, all these little fragments laying around, and they're not just acting in isolation, they're actually acting together to treat infections. And then the other thing that we did was that I thought might be interesting to share is that, of course, when you're trying to develop a new antibiotic, you want to make sure that the target doesn't readily develop resistance to your antibiotic. And so the target in this case is bacteria. And so here I'm showing results from a resistance development assay where we continuously expose, in this case, this gram-negative bacterium, A. baumannii, to a conventional antibiotic, polymyxin B, that is used in the hospitals, and 3 of our encrypted peptides. And we can see that the bacterium eventually develops resistance to polymyxin B, but not to the encrypted peptides, at least within the constraints of our experiment, which was a 30-day experiment. And so this is also encouraging results where you want to minimize resistance development if you're trying to develop a drug. And so this was also encouraging. And then we tested the anti-infective efficacy of these encrypted peptides on 2 different mouse models. These are the results for the skin infection model. We were able to also recapitulate the synergistic interactions that we observed in vivo, also in vitro. So these are monotherapy treatments with one encrypted peptide or another encrypted peptide, and this is the combination of both of them. And then this is the untreated control group of mice that are infected with Pseudomonas aeruginosa, this gram-negative pathogen. And similarly, these are mice infected with a different bug, but it's the same concept. So untreated control group, monotherapy, and then combination therapy. And then we also tested them in a deep thigh infection model, which is typically the one that the FDA wants to see for any sort of preclinical leads. And so these are some of the results. This is the untreated control, and again, monotherapy, and then this is the combination therapy. So it seems like when you combine them in cocktails, you do get a potentiating effect that could potentially be interesting when thinking about in the future translating some of these concepts into the next step. This is just to show you that at least four of the encrypted peptides that we found, they're actually produced physiologically in the body. This was we did this through a literature search, looking at prior literature, and we can see that they're produced at different levels, from low to medium to high, and in different areas of the body. And so this just brings further support that maybe these things are actually playing a physiological role and an immunological role in our bodies. Okay, I'm going to shift gears a little bit, but I just wanted to mention quickly another project that I thought it would be interesting to share, where thinking about antibiotics, right? They're very passive molecules, right? Let's say you inject them in the bloodstream, and they just move along the bloodstream with no direction whatsoever. They're essentially not targeted to the site of infection, right? And so this brings about a lot of side effects, unintended effects, where it's toxicities, or you can kill your good microbes and so on. And so we started this collaboration, and we're able to initiate this concept of autonomous antibiotics, where there's essentially self-propelled to the site of infection. And so what we did is we used these nanomachines, and we loaded them with a peptide antibiotic. And the nanomachines are capable of self-propulsion through a distance. And what they do is they transport the antibiotic, and they release it as they go, clearing the infection. And so I'm going to show you a little video. In this quadrant here, you won't see much because the substrate that drives the reaction has not been provided. So you'll just see Brownian motion. But in the other quadrants, you'll be able to see self-propulsion of the nanomachine, directing the antibiotic to the infection site. And this is in vitro, but we demonstrated this concept in a mouse model as well. So anyway, this is just, I thought I would share. It just opens maybe new ways of thinking about how to deliver drugs to the site of infection, thinking about personalized medicine and so on. Okay, so just to summarize, so we're very excited about the potential of computers to help us design and discover novel antibiotics or novel drugs in general. Now we're working on trying to streamline that to a robot that makes the molecules the computer tells it to make, and then a screening facility that screens those molecules for antibiotic activity or other types of activities. And then those bioassay functional data, where we'll be able to build models to tie them back to the computer, so then we can have this sort of self-learning platform to come up with novel antibiotics or novel drugs. And just to illustrate how young this field is, at the intersection between AI and antibiotic discovery, this is a retrospective study that we run looking at publication results. And we can see that papers having the queries at the intersection of antibiotics and AI, they only started going up really in the year 2018. So this is an extremely young field. And so I always like to encourage the young audience to try to join these efforts that we have. It's a highly interdisciplinary field. You can have people from physics, from chemistry, from computer science, from biology, all working together. And if there's anybody out there that is interested, I would encourage you to join this important effort. And yeah, I just like to a little plug here. American Chemical Society, they usually ask me to add this to the presentations, but this is a book that we released recently, and I think it's an interesting primer for students trying to get into the field. So if anybody's interested, please reach out. I may be able to get a copy. And with that, I'd like to thank my lab members that did a lot of the work that I've presented, our great collaborators and funders that allow us to do the work we do. And here I'll just leave you with links to our website, email address, and Twitter handle, in case anybody wants to get in touch through any of those means. Always happy to connect. And again, thank you so much for having me. Thank you for your attention, and I'd love to take any questions.